lecture for the Teaching Excellence in Entomology program. And those of you are probably very familiar with Yahel's role in teaching. He's pretty much been a permanent TA, working with me for the entirety of his degree. It means that he's got to do a lot of innovative things that he wanted to do in teaching, like the structure and function labs are really different now than they were before because he has a lot of crazy types of experiments. He has a really unique teaching style that I'm excited for him to get to showcase today. And his syllabus, which is on the email, you can look through. And undergrads, if this is something you want to take, you should petition for him to be hired as a lecturer.
Whoa, didn't expect that part.
Yeah, you did.
Not yet.
I wasn't here. It's not me. Hello, everybody. So as Chloe said, Dr. Hawking said, this is part of a course. So this is like, I think, the second or third lecture within the course. What? And this course is aimed at undergrads, which most of you are not, but so you know. It's about the diversity of plant chemistries in nature. So let's start this off. All right. What is this?
Tomato.
It's a tomato. Oh, you're clever, aren't you? All right, what else do we see? You see roots, okay, and soil. We see roots, we see soil, we see leaves, we see fruit, we see flowers, right? What else? Any other observations?
Blooming.
It's blooming, yeah. It's also green, right? Tomatoes are red. We see a lot of things here. The plant is in the ground. So then the big question is why? Why are all of these things the way they are? Why is the plant green? Why are there flowers and fruits and roots? You know, what's happening? So because this is a chemical ecology course, we're going to talk about plant chemicals in this lecture. And there's two overarching groups of plant chemicals. For those of you who don't know plants, welcome. The first group we call them primary metabolites. And the second group we call, that's right, secondary metabolites. Whoa. And so what are primary metabolites? These are any chemical within the plant that is directly necessary for normal growth, development, and reproductive functions of the plant, right? So this could include many of the fundamental proteins, carbohydrates, and lipids. Those are the three basic polymers for most of what goes on in the plant. And most of these compounds are derived through photosynthesis and respiration. And, you know, they include lots of amino acids, lots of sugars that build up the plant. And in the context of herbivores, they love them, right? That's what they eat. You know, they need those nutrients. And other section of primary metabolites within a plant includes the hormones related to the functions of normal growth, normal development, reproductive functions. And so some phytohormones that are considered primary phytohormones are auxins. These are a class of compounds that affect root growth, apical dominance, and phototropism within a plant. We also got cytokinins, which these affect how the shoots grow and how the cell divides and differentiates within the plant, right? So we got growth and development. We got gibberellins, also major growth hormone for plants, and it promotes flowering and breaks the dormancy of the plant. And abscisic acid, which is kind of the opposite of gibberellins, which this enters dormancy. It helps the plant enter dormancy, closes the stomata of the plant. And lastly, we got ethylene, which is for fruit ripening and leaf abscission. So now that we go back to this plant, right, we have the plant growth. The fact that the plant grew is because of the primary metabolites, why it grew, why it developed, the different parts of it, the reproduction, the fruits and the flowers, all as a result of primary metabolites. It made the plant green, right? Because if it's related to the primary functions of growth and development, that means it's also related to the photosynthesis, which requires the chlorophyll, which is green. So that's all primary metabolites. But it's still stuck in the ground. So what do we do about that? If you're a plant, if you're stuck in the ground, what can you do? Anybody. Defend yourself. Whoa. So there's a whole other groups of phytohormones which are not directly for the growth and development of the plant, but for other factors, such as gysmonic acid. This is a big one. And it's used often in plants to defend against chewing herbivores and necrotrophic pathogens. And then on the other hand, we have salicylic acid, which defends against sucking herbivores and biotrophic pathogens. There are a couple other phytohormones, such as strigolactones, which, while this isn't directly defense, these guys are phytohormones that promote mycorrhizal growth, which is symbiotic fungus. And so when we think of these three phytohormones, they're not helping directly with the growth or development. They kind of require almost something else to be there, right? Especially the salicylic acid and the gysmonic acid phytohormones, they're defense mechanisms. There's going to be more of them if they're being attacked. Similarly, the strigolactones go through the root exudates, meaning they're not helping the plant, they're more so attracting something else. And also ethylene, what we talked about before, about the ripening. This is actually a gaseous hormone. And so what that means is that it could communicate with others. If you ever put like a tomato in the same bag as like an avocado or a banana, they help each other ripen. And that's not necessarily like they will eventually do it themselves, but they help each other ripen. It's a gaseous, there's communicating with other organisms here. And so this brings us to the next group of metabolites, the secondary metabolites. These are the compounds in the plant which are not directly associated with the primary metabolism. And these were originally thought to be waste products, but now we know that they're not waste products. They are more than just waste products. These can be attributed to defense mechanisms within the plant. They could be attributed to resistance against environmental stressors and between communication, whether it's communication with other species, with the same species, different organisms. And this is what chemical ecology is all about. It's all about these communications, these interactions between different organisms by use of chemistries. And secondary metabolites are very diverse in their structure, in their function, in the biochemical pathway, the location within the plant where they are. Here's a little flow chart where we have photosynthesis at the top, which creates carbohydrates. And there's a whole load of different ending secondary metabolites which result from this. It's crazy. People used to think these were just waste products. And these are often simplified into three categories. There's more than three, but three basic categories of the secondary metabolites. We got nitrogen-containing compounds, terpenoids, and phenolics. So when we look at this chart, we got a whole area that derives from the amino acids, which are nitrogen-containing compound, which makes sense because amino acids contain nitrogen. And we got also a whole section here, which are the terpenoids. And if you added nitrogen to a terpenoid, then it could be multiple. There could be overlapping. And then the rest here are phenolic compounds. And if you look at these real close, some of these are also including some of our primary metabolites that we talked about. Like down here, we have abscisic acid, right? We got the cytokinins and the gibberellins. These are also different compound and chemical structures that could take different forms. And if we look at it in a different way, we got the photosynthesis that kind of leads to carbohydrate metabolism, fatty acid metabolism, and nitrogen metabolism. Remember, these are our primary metabolites. We got lipids, right? The fatty acid, we got the carbohydrates, which is like sugars and starches, and nitrogen, which is associated with amino acids. And these could go on to lead to multiple different secondary metabolites, which fall under these groups. And there are so many of these metabolites out there. And we went from metabolites to secondary metabolites to nitrogen containing compounds. And within each one of these groups, there are further classifications of other compounds. And we're going to go into some of them. So the first compound that we're learning about today are alkaloids. Who here has heard of an alkaloid? That makes most of us, right? So these are cyclic nitrogen-containing compounds. So here's a couple examples of cyclic nitrogen-containing compounds. And in how we pronounce it, most of them end with the suffix "in." So does anybody know any? Caffeine, what? Any of you drinking your coffee now? You're drinking alkaloids. Whoa. We have nicotine. Yeah, nicotine. Theobromine, that one's in chocolate, if anybody. All these good stuff, right? All of these good stuff. These have a strong effect, whether they're positive or negative on us, whether they're positive or negative on different insects and different plants. And so when you actually delve into the chemistry, most of these are weak bases. They're not very slightly soluble in water, and they're generally bitter tasting, right? So all of you drinking your coffee, all of you drinking your chocolate, they all have a slightly bitter taste, and that arises mostly from these alkaloids. And when we think about this in the context of chemical ecology, in the context of a plant trying to defend itself, many of these are insecticidal or toxic compounds. And they're found in many plant families. Usually each alkaloid is kind of specific to each plant. The Solanaceae family, those are like tomatoes, nightshade. Those are well known for having a lot of alkaloids. And interestingly enough, if you take one of these alkaloids like indole and you slap an acetic acid group on it, you get oxen, which is one of our primary metabolites we were talking about. And so if we look at here, right, we have different plants that can produce different alkaloids, right, in their own ways. Also, debatably, some people can debate that DNA is an alkaloid because it contains that cyclic nitrogen group. However, DNA, as we know, the A is acid. Alkaloids are generally weak bases, so people don't always associate these guys with alkaloids, but it depends who you ask. Our next group of nitrogen-containing compounds are cyanogenic glycosides. So when we say glycoside, it means that it comes with a sugar molecule. In this case, you'll see glycoside a little more later. And these are nitrogen-containing compounds that contain a cyanohydrin group, which is this here carbon triple bonded to nitrogen with a hydroxy group that is taken by a sugar. And there are a couple chemicals here, like prunicin, amygdalin, and linomagrin are three very well-known cyanogenic glycosides. And that one's samburanagrin. Most of these are named after the plants that they were found in. Fun fact, like prunicin is from prunus, which are cherries and peaches, and more. And so how these guys work is they are very toxic, so toxic that the plant can harm itself if it has them. So they're stored in inactive forms within the vacuole, which is an organelle, which is for storage. And when it's damaged, it releases this cyanide group. And that's very toxic. It's toxic to the plant. It's toxic to the thing that eats it. And so many plants can metabolize these. However, it's very rare for a plant to accumulate them because it's so dangerous also for the plant to have it. Rosaceae, these are the apple family, are well known of being able to accumulate it. And if you ever heard of that bitter almond smell, that's how these guys often smell and taste. For those of you who are interested in tasting these very toxic compounds, right? So they're found in like the almonds and in the apple seeds, right? We heard that they have cyanide. That's coming from the cyanogenic glycoside. It's also found in cassava, fun fact. And our next nitrogen-containing compounds, we have our glucosinolates, which these are nitrogen-containing compounds that have a glucose, which is a sugar, and a sulfur molecule. And some very well-known compounds of these are synegrin and synalbin, and also glucotropaelin. And these are a pretty conserved group. And how their mode of action works is that there's this enzyme called myrosinase, which comes and hydrolyzes the glucosinolate into this bad boy, this isothiocyanate. Notice the cyanate again. Pretty toxic and unpalatable, especially to generalists. However, there's many instances in which a specialist has turned that around and been able to sequester and utilize. But for most generalists, these are toxic and unpalatable compounds. And they are mostly found in brassica, which are the mustards. And so if you ever want to taste this, these are the mustard oils. If you ever smell some mustard, that's coming from your glucosinolate. And they're found in many brassicas and also papayas, which is fun. And so now that we have a lot of these very toxic nitrogen-containing compounds, how can we exploit them as humans? Now if we know about it. So we know that these are very toxic, and people have made many insecticides out of nitrogen-containing compounds because of their high toxicity, such as the group 4 pesticides. These are neonicotinoids. These are all alkaloids, and they lead to death of the insect. There's so many other pesticides out there which have nitrogen in them, whether they're alkaloid or not. And so these are some ways that we could use alkaloids and other nitrogen compounds. If we know that they're toxic, how we could use them for our pest management, for instance. Our next compound class that we're going to go over are terpenoids. So these are made of isoprene units, which is this. It's a bunch of carbons. There's a couple double bonds. This is one isoprene unit. And when we look at the context, there's so many of these compounds that have been characterized, and they could be acyclic, they could be cyclic or bicyclic or more. And they are often named after the amount of the isoprene unit, right? So a monoterpene has two. It's a little tricky. A monoterpene has two isoprene units, but you see 1, 2. Sesquiterpene is 3. And there's a lot of different plant products that have these terpenoids in them. And they have the different functions within the plant. So we're going to talk a little about monoterpenoids. These tend to be volatile in essential oils. They are pretty small. There's only 2 isoprene units within them. So they volatilize, and then you could smell them. And so they need often very specialized storage cells to be able to contain them. And they include some of these nice smells, right? Like the geranium smell, catnip, pine smell, good stuff. 2 units of isoprene. Does anybody want to come up and try to find them? Yeah, yeah. I'll show you what an isoprene looks like again. This is an isoprene. All right, I'll do the first one. Right? We got one isoprene unit, another isoprene unit, right? Easy, easy. Who wants to do the next one? Come on over, yeah. So these smell very nice for us. They could also smell bad for us, but they smell. That's a good thing. All right, all right. Which one do you want to do? 1, 2, 3. Yeah, so if we, yeah, right? So we see this isoprene unit here. Looks just like this, exactly the same, right? And then another one on top. And for this one, we have the same situation, right? So if you ever see a compound like this, you're like, that's a monoterpene or a monoterpenoid. Fun fact, different. I don't know why I say fun fact. They're not always fun. But monoterpenoids versus monoterpenes, when we hear terpene and terpenoid, the difference is the ones with oids have things other than just the carbon, such as this has here, this OH group that makes a terpenoid versus the terpene, but they are very similar. Thank you, Alex. So the next one are sesquiterpenes. These are also often in essential oils, such as farnesene and humulene. Anybody like beer? This is what the taste of hops comes from, the humulene. Farnesene is also very commonly found in many plants. Do we see the isoprenoid units, right? The isoprene, right? Yeah. You're not. You want to come show us? You want to come? Come on, Emma, you got this.
Help you out by nodding.
Yeah.
I have to participate.
Hate participating.
There's one.
Yeah.
There's
What?
there's
Whoa! Round of applause for Emma. Also, round of applause for Alex for before. Yay! Anybody want to try the humulene? It's a little trickier. A little trickier. Nobody wants to try the humulene, huh? And you want to try the humulene? No, you do not want to try the humulene. All right, I'll just show you. Ooh, a couple more information about this first is that these are very important in olfactory use for insects, very common in Asteraceae, and sesquiterpenoids can include and lead to some of our other primary metabolites, such as abscisic acid, strigolactone, and gismonic acid. We see the isoprene unit. We see the isoprene unit. Yeah. And here's Emma's isoprene units over here, right? Easy. We got this. Isoprenes. Love to see it. Sesquiterpene, 3 terpenoid isoprene units. Brings us to the next one, diterpenes. 4 isoprene units. It's crazy how life works. And so when you're getting bigger in the molecule, it's becoming less and less volatile. The molecule is heavier, harder to be out in the air, lower vapor pressure. So these are more common in latexes and resins. That's a way solider substance than like a volatile. And here are 2 anybody is interested in up for the task of finding the isoprene units. It's kind of a little puzzle. It's kind of fun, isn't it? Stephanie, I see you counting. No, okay. And so if we look at the actual function of these big molecules, or bigger molecules rather, like in latexes and resins, these cause a lot of feeding deterrence in insects. And these also include some of the primary metabolites like gibberellins. But so we have here each isoprene unit, right? Beautiful. The next one. It's only going to get more complicated. We ready? Triterpenoids have six isoprene units. So they include this bad boy like squalene. Pretty simple, right? But fun thing about squalene is that then becomes squalene is the precursor to steroids, which are a form of triterpenoid. Um, and these steroids there and triterpenoids are common in latexes and resins and can include many, many compounds. I'm not going to ask you to try to break it up, uh, but just take a look. By the way, steroid is what we call a molecule that has this shape with the six rings and not the six ring, the four six carbon rings followed by one, the three six carbon rings followed by the one five carbon ring. These are steroids, and these can be found as triterpenoids within many plant compounds. So it's just cucurbitacins and liminoids. Those are two defensive compounds that are very deterrent in feeding. They tend to be toxic. Uh, Phytoicdisteroids, we heard about icdisteroids, right? Icdices, right? Um, that's a steroid that comes from a triterpenoid. And that leads to um, it could mess with the molting of and metamorphosis of an insect. We also have saponins and cardiac glycosides. Remember, glycoside means they're attached to a sugar. Here's a cardiac glycoside. It's attached to a sugar. And that could cause heart attacks, cardiac, um, found in many like milkweeds, for instance. And so these compounds tend to be pretty deterrent in feeding. They're large, they're in resins that could be toxic. Um, and they're only even getting larger, these terpenoids. So some terpenoids are so big, such as carotene, which is a tetraterpenoid, eight copies of isoprene units, and even bigger becomes rubber, uh, which is a polyterpenoid, which is just lots and lots and lots of isoprene units. The bigger they get, the heavier they get, the more rubbery they get, but they could also be pretty toxic. And so when we look at exploiting terpenoids, we got many toxic compounds, right? So one of the most common insecticides that we have based terpenoids off of are pyrethroids. They keep the sodium channel open, leading to hyperexcitation, leading to a nerve block. These are your IRAC3 groups. And also juvenile hormone mimics, which people have used. These come from terpenes because remember, we said phytoicdisteroids. These are steroids produced by the plant, which mimic or they influence the metamorphosis of an insect. So in these cases, these are terpenoids that have they're mimicking the juvenile hormone, and they can be applied to young insects and disrupt metamorphosis. And so for our next overall grouping, phenolics, everybody's favorite. I could tell by all of your faces, you love phenolics. These are very widespread amongst plants. And they, similarly to terpenoids, are classified by the number of carbons found within the plant, within the compound. So there's this fun little table. Number of carbon atoms is how we classify them. With the most basic one, we call it a phenol, right? A phenol, this is the namesake of phenolics. It has this benzene ring, has this hydroxyl group. It's considered a, anybody want to count how many carbons in there? How many carbons are in there? Six. Yes, we can count. You could also look at the chart if you didn't feel like it. This is the most basic form of a phenolic called phenol. Makes sense, right? You get a little bigger, like salicylic acid, which was one of our phytohormones. What do we see here? We see how many carbons? 7. And how many are in the ring? 6, right? Because that's how a phenolic structure is. So we have that 6 carbon ring, and we have a 1 extra carbon, and these are called C6, C1, because it's based on the size of the carbon, the amount of carbons. And then if we get even bigger, like coumarin, how many carbons do we see? 9. And so how do we think we're going to call it? Yeah, we call them C6, C3, because all of the phenolics are going to have this phenol group, right? Which is the benzene ring with the oxygen. And then any other carbons coming right off of it is what we call the extra backbones. And so then you could build more phenol groups off of this. And one of the most common groupings of these phenolics are called flavonoids, which are C6, C3, C6 compounds. And this is debatably the most diverse group of phenolics that we have, and they're also often stored in vacuoles, which is within the plant, and they could include these different compounds such as catechin and rutin, C6, C3, C6. Anybody wanna try to point one out? Yeah, you're all avoiding eye contact with me. Okay, okay. So here we have, right, C6, C3, C6, right? We got this phenolic group, followed by 3 more carbons that are directly attached, followed by another phenolic group that's directly attached. That's the catechin. We also have rutin, and if you look at rutin, it's attached to some sugar molecules. So depending, all of these can overlap with each other in some ways. And so these can be feeding deterrents. They could be feeding stimulants if the insect is adapted to it and flips it on its head. They could be toxic. Also, they could include some pigmented anthocyanins, which are also antioxidants. Phenolics are responsible, in general, phenolics are often responsible for a lot of flavors. Like if anybody's ever had blueberries, we love blueberries, right? Full of phenolics. Within the berry, within the leaf, there's many phenolics out there. And then that brings us to tannins. So these are large polyphenolic compounds, such as this bad boy. We see in this specific case, this is a large compound made of flavonoids, right? Because here is one kind of flavonoid group of the C6, C3, C6. They don't have to be, but many of them are. And they could be hydrolyzable or condensed. This is an example of a condensed one. A hydrolyzable would be one that can be hydrolyzed, which means broken apart using water, usually hotter water. And these are very common in many plants, and they bind to proteins and they become indigestible. So in the context of a chemical ecology, if these bind to proteins and become indigestible, what do you think the use of them for a plant is? Yeah. Yeah, exactly. Preventing insects from eating unripe fruit. It doesn't only have to be fruit. A very popular plant that has a lot of tannins in it, for instance, are oak. If you look at oak leaves, they have a lot of tannins in them. People who love wine are going to know about these tannins also. They're responsible for that dryness, that astringent flavor. I guess it's not a flavor so much as a texture situation. Because it binds to the proteins, it kind of takes it out of your mouth. It's also widespread amongst plants, and becomes difficult for the insect to eat it. Obviously, you're going to have insects out there that are specialists, and such as if we look at the oak example again, one very big pest of oaks would be the spongy moth, and those kind of like the tannins at this point because they eat so many. But against most herbivores, it's gonna start deterring them. And we could exploit phenolics in our pesticides because many of them are feeding deterrents. However, because many of them are feeding deterrents, they're not directly toxic like some of the other ones. So there's way less phenolic pesticides out there than like the nitrogen containing pesticides. But one phenolic pesticide that is actually derived from a naturally occurring phenolic is rotenone, which is a mitochondrial complex electron transport inhibitor. But there's many phenolics out there and are used by the plant to kind of deter the insect from feeding. And so overall, there's many factors that could affect the chemistry of the plant. There's the location within the plant. These are all things that will be talked about in future lectures in this class that I'm proposing. Within the plant, there could be varying concentrations. If you look at a plant like the tomato plant from before, the flowers were yellow, the fruit was red, the plant was green. Different parts of the plant have different compounds in them at varying concentrations. Different ages of the plant could also lead to different plant chemistries within the plant. A young plant versus an old plant might have a completely different chemical profile. Maybe not completely different, but relatively different, and especially with the concentrations involved. And compartmentation. So we talked about the vacuoles, right? Such as some of those very nasty compounds, like the cyanogenic glycosides have to be held in a compartment or else they harm the plant. Many of those monoterpenes are very volatile, so they need special compartments like glandular trichomes to hold within them the compounds. So within the plant, things can affect the plant chemistry. We also have the environment of the plant, which includes basic things like the sun or the shade. You have, well, we're talking about some of these volatile compounds. When heated up, they become even more volatile. If they're in the sun, they're going to become smellier, whether that's a good thing or a bad thing. The time of the day, the season, right? We talk about how abscisic acid is responsible for closing stomata, entering dormancy. That's not going to happen in the summer, most likely, for most plants. They might close their stomata in the summer because it's hotter, but they're not going to go into dormancy. It depends on the time of day or the time of the year. And there could be various soil factors as well. And here's a very nice graph I took from this paper, where if you have a plant producing all of these fun secondary metabolites that we talked about today, right? The phenolics, the terpenoids, the alkaloids, and they're all influenced by these environmental parameters. And of course, insects. So within a plant, you could have induced compounds, you could have constitutive compounds. We're going to talk more about it in a different lecture, but essentially what that means is if you have a plant, it has a lot of tannins, for instance, it's gonna just have tannins all year long. That's constitutive. It's always there. It's not induced. It doesn't come up from the damage. You could have other things though, like some of the terpenoids or salicylic acid, for instance. If you have some insect damage, it's gonna trigger the pathway which is why it's a hormone, to release some compounds. And in this case, it's as a response, as opposed to being always there. And there's different theories of why some compounds might be induced, some compounds might be constitutive. There's different theories of where they are in the plant, why some compounds are in certain areas versus other areas, how the plants utilize them over time. An older plant versus a younger plant. Why does a plant chooses anthropomorphizing it, but why does a plant choose to use a certain compound in a certain location at a certain age, with all of these different factors? When you consider one plant, a whole lot of plants, one herbivore, a whole lot of herbivores, the different trophic levels, this gets really broad. And so knowing the different plant chemistries can help build kind of a foundation of knowledge when we get deeper in this course. It also will help you actually be able to identify these different compounds. Like if I show you one of these, you're all gonna be like, that's a phenolic compound. I can tell that's a phenol group, right? And obviously also genotypic variation. Different plants are different individuals. Different individuals might have different genotypes, even if by slightly, and will express different genes to on slightly different levels that can lead to more or less of certain plant chemistries. That's why you could have kale that is green and kale that is purple, right? They're both kale, but there's something in them that is making one more green, making one more purple from a breeding point of view of the plant. And so for the next class on the syllabus, we are going to talk more about the theories of the plant defenses. And yeah, make these chemicals for defense, for all of these purposes. Humans can use them for all sorts of products. And then there's some unexpected effects. And those are what I have for today's lecture.